Cell Structure Part 4. The endomembrane system consists of membranes and organelles and eukaryotic cells that have very important functions in terms of manufacturing materials that cells will need to grow and metabolize and replicate, make copies of themselves. It's a large factory. In fact, the cell as a factory or the cell as a city is often an activity that's given to elementary school children to understand the complexity of plant and animal cells, of all of the different components that must work properly together for a healthy cell. It's all about the cell. If the cells are healthy and functioning properly, then the whole organism itself benefits. The parts of the endomembrane system include the nuclear envelope, all of the endoplasmic reticula, smooth and rough, the Golgi apparatus, lysosomes, vesicles, and the plasma membrane. So it's a transport system and a factory system to manufacture all the materials and transport all the materials needed inside of a cell. This illustration tries to show you that sort of continuous arrangement and continuous association, starting with the nuclear envelope, transitioning to the endoplasmic reticulum, then the Golgi apparatus, including various different kinds of vesicles and lysosomes, and the plasma membrane. And this is constantly happening in your cells. Certain types of cells more vigorously than others. See if you can label all of the parts. Practice labeling these structures, since this will be one of the types of activities you might be asked to do in an exam in general biology. You might also see this similar kind of illustration if you're taking another type of course in biological sciences that discusses cell structure. For the endoplasmic reticulum, we have the rough and the smooth. Here we have an example in the bottom image. It's a transmission electron micrograph of RER. On the right is smooth ER, and on the left is RER. So we have rough endoplasmic reticulum on the left in this TEM photo, and in the upper right is some smooth. And you should be able to easily see the distinguishing feature between the two visually is the absence or presence of ribosomes. That's the reason scientists gave these names to these structures. One appeared rough, the other appeared smooth. We also see the association with the nucleus and the nuclear envelope, and we even have an arrow pointing to a nuclear pore, which isn't always easy to find in these TEMs of cross-section of a cell. If we look at the structure of endoplasmic reticulum, the internal structure is called the cisternal space, sometimes called the lumen. The cisternal space or the lumen is the location into which chemicals and materials, biochemicals, that are manufactured through the surface of the endoplasmic reticulum is deposited. So materials, biochemicals, are manufactured on the surface of these endoplasmic reticulum structures and deposited into the cisternal spaces or the lumens. For RER, the material that's manufactured and transported into the lumen or the cisternal space is called protein. Proteins are important for many tasks inside of a cell. We find that phospholipids are also synthesized in the RER, and phospholipids and proteins may need additional modification. If they don't need any modification, then they're going to be transported in what we call vesicles right straight to the membrane structure of the RER. These vesicles may be transported, if they need further modification, to the Golgi apparatus. So constantly, as proteins and or phospholipids, especially the proteins, are being manufactured and dumped into the lumen or the cisternae of the ER, rough ER, they get packaged in little vesicles and might need further modification. And those are going to get transported right to the underside, what we call the cis face, of the Golgi apparatus. And then step by step, inside the Golgi apparatus lumen, or the internal surfaces, those modifications occur. And you can see there's a transition from the inner surface of the Golgi through various stacks, or through various transitions, to what we call the transface, the surface of the Golgi that's closer to the plasma membrane. Eventually, at the surface of the Golgi that we call the transface, additional vesicles are released, sometimes called transport vesicles or secretory vesicles. These secretory vesicles now contain the further properly modified, fully functional structures or biochemicals in the form of proteins packaged inside. Those vesicles can fuse with the plasma membrane. Once they fuse or blend in with the plasma membrane, then the chemical proteins can become embedded in the plasma membrane. And that's what's illustrated in this little cartoon drawing. For SER, the SER and the RER have continuous arrangement. But again, the SER tends to be viewed as having no ribosomes on the surface, or maybe just a few. And the materials manufactured in the SER are different than the RER. Inside the smooth endoplasmic reticulum, we find the synthesis of carbohydrates, lipids, and steroid hormones. There's some detoxification of medications that can occur in the SER, and the detoxification of what we call poisons can also occur. For certain types of cells, there's a specialized SER for the storage of calcium ions. Those types of cells that need specialized SER are muscle cells. And the name given to the SER in muscle cells for the storage of calcium is sarcoplasmic reticulum. We need calcium for the coordination and contraction of muscles. And in fact, you can see in this illustration of a muscle cell, often called a muscle fiber, that the SER has a very complex, unique kind of structure that's what we would expect to find in a muscle cell, and not the idealized or stylized animal cell that we've been seeing in our previous illustrations. The Golgi apparatus, again, is that further step in the modification of proteins and possibly lipids, like phospholipids, that still need additional attachment structures added to their biochemical structure. And when you see a Golgi apparatus, you can't mistake it for anything else. They're generally cup-shaped or a little bit curved on one surface, so they are essentially going to have sort of a concave, convex appearance. And you can see in some of these static photos, like the top transmission electron micrograph, where we can see vesicles and we can see structures seeming to become aligned to the undersurface, or the cis face, the surface of the Golgi that's closest to the nucleus and closest to any endoplasmic reticulum, compared to the little vesicles that are blebbing out on the outermost surface of the Golgi. And we see Golgi as sort of these pancake-like structures. There's very sort of interesting, again, slightly curved stacks of membrane structures. So once you see a Golgi apparatus in an illustration, and then you see one in a real image from a cell, like this photo at the top right, you can't mistake it from anything else. So the surface closest to the nucleus and closest to endoplasmic reticulum is the receiving side. And there's going to be a transport vesicle, a membrane-bound vesicle, that's going to bring biochemicals, usually proteins, to the cis surface or the receiving side of the Golgi. Deposit those chemicals. It's like a factory. So we're going to deposit the building blocks and the materials to this finishing factory. And then the biochemistry occurs in the lumen of the Golgi, in the spaces inside the Golgi, sort of little flat membrane structures, until we get everything processed and properly formed, and you get to the transsurface, which is the secretory vesicle surface, and now these finished products can bleb out and migrate to the plasma membrane.